Hello, welcome. Thank you, everybody, for joining with us today. We're broadcasting live from Park City, Utah, where I am. Kevin, you're in, remind me?
Newton, Massachusetts.
Newton, Massachusetts. Brent, you're in
Right
a big
next door.
conference. You're in a big conference from somewhere. Yeah, Dave. We have Kevin, Brent, and my friend Dave, who has introduced us to Lumicell, and I just want everybody to know that it's all his fault. So you'll know that and discover that he's super active with this company as an investor, and we'll have him talk a little bit about that later. So again, thank you for everybody for being here today, and big thank you to the Family Office Insights community for being as active as you are. As you know, we can't hear or see you. We know you're there, and I would encourage you to put your questions in the chat box in the Q&A. We'll get to as many as we can. Today, we're going to be doing what we call a Luminary series. We're going to be talking about what's going on in the industry, and in particular, how that applies to the company here. And then in a week or so, we'll be doing another official pitch webinar, so just stay tuned for that. And as you listen to our webinar today, if there's anybody that comes to mind that might benefit from hearing what you hear, we are going to record it, and we'll also make sure that you're in direct contact with the company so you can chat with them at your liberty. As you know, we don't stand in the way of that. So with that, again, thank you so much for being here. Kevin, you want to start us off?
Sure. Thank you, Arthur, and thanks for everyone for joining today. Very happy to be here. My name is Kevin Hershberger. I'm the CEO here at Lumicell. My background, I've been a range of medical device companies, Boston Scientific, Next Stage Medical. I did a startup, Rewalk Robotics. And I've been here at Lumicell for about 4.5 years. I have to say that in my career, in all the technologies I've had exposure to, this is a technology that Lumicell has a technology that has the opportunity to really impact a lot of patients. Our first indication is breast cancer. And what we're doing here is really important. We're addressing one of the largest issues with cancer surgery, in that during the initial cancer surgery, there's typically cancer left behind. Clinical studies have demonstrated that surgeons leave cancer behind because they can't see it. They know where the primary tumor is, but there's cancer left behind. And we've developed a solution that actually helps surgeons find cancer that's missed. Our first indication is breast cancer. And we are very happy to say that our clinical trial results were just published in New England Journal of Medicine Evidence that demonstrates we're finding meaningful tumor in these patients. So, that's a quick overview. I'm sure we're going to get into a lot of Q&A later, Arthur, but I'll turn it over to Brent to introduce himself.
Hi, good afternoon or good morning, depending on where you are. My name is Brent Palmisano. Like Kevin, I've spent the vast majority of my career in medical technology. I'm approaching 20 years at Boston Scientific, predominantly the majority of that time, and then a shorter stint at another late stage startup, and have had the privilege to be here at Lumicell for the past year, and the distinct pleasure of leading the commercial efforts once we get approval from the FDA. Thank you.
So is it I'm going to take a crack at giving a sort of layman's description of this, just because I think it might be a good starting point. So Kevin, you mentioned it's a way to identify for the surgeon to remove, I guess, tissue that they would otherwise miss. And that the science behind this is you have some sort of light, I'm going to get this all wrong, that helps them identify that, and so you don't have to go back in and remove it again, and there's a higher probability that it doesn't recur. Is that not bad?
That's pretty good, Arthur. That's great. And I can fill in some gaps. I mean, so put it in perspective. There are about 2 million new cancer cases in the US alone every year. More than half of those are solid tumor cancers. So the objective when treating a solid tumor cancer is to remove the tumor first. So no matter what you do before or after surgery, the primary intervention and the primary focus is to remove that cancer completely, complete resection of that cancer. So breast cancer is our first indication. And that's a great example to use because the entire care continuum in treating a patient for breast cancer is about complete resection. And it starts when they diagnose, when they biopsy the tumor, when they localize the tumor before surgery, and then after surgery, they rely on margin pathology to determine if there's tumor remaining on the edge, on the margin of the tissue that's been removed. And that implies that if there's a positive margin or tumor on the edge of that tissue that's been removed, it infers that there's tumor left in the surgical cavity.
They might have missed something. Yeah.
Correct. The reason that's so important is that incomplete tumor resection, so tumor left behind, doubles the risk of recurrence. And by the way, there is a 25% mortality rate when there is a recurrence in breast cancer patients. So what we do is, what we've developed is the first of its kind solution that allows surgeons to look inside the surgical cavity. There are a lot of technologies that have been tried over the years to improve margin pathology. So looking at tissue that's been removed, okay? But until now, surgeons haven't had the ability to go in and look inside the surgical cavity. We have a fluorescent imaging agent that is activated by the body's host immune response to cancer. And that's important. I know that it's activated because the mechanism of activation is what makes it a pan-cancerous solution. So the fluorescent agent is activated by the tumor, and the tumor's the body's host immune response to tumor, and it allows the tumor to light up. It fluoresces the tumor and in the tumor microenvironment, so it essentially creates a halo effect around the tumor, so the surgeon knows exactly what tissue to take, so there's nothing left behind. We combine that with an imaging device that goes inside the surgical cavity and views and images any tumor that's left behind. And if the third leg of the solution is a tumor detection software, which is calibrated for that individual patient. So it takes in, I mean, it essentially takes into consideration any autofluorescence that patient may have.
So if to dumb it down a little bit for me, is the illumination of the tumor and the surrounding area happening during the surgery, before the surgery? How does that work?
So, that's a good point. So the agent is administered to the patient 2 hours before surgery. Okay, so it's 2 hours. It's systemic, so it gets through the body, and the surgeon already knows where the primary tumor is, so they're only imaging for residual tumor. They're not imaging the primary tumor. They're going into the surgical cavity and imaging to see what's left behind. And it's important. Okay, go ahead.
But then what happens?
Then the surgeons know exactly, the surgeons take out tissue that's been left behind. So there is on our website, there's an animation, and there's also a video that shows Dr. Barbara Smith, who is the head of the breast cancer program at Harvard Medical School and Mass General Hospital. And you'll see in that video, what she's doing is she's removed the primary tumor. She scans the cavity. She lights up tumor. So it actually shows the screen that it says positive, and it shows the red signal. She leaves the device in and puts forceps in so she can grab a hold of that tissue that needs to be removed. She removes the cancer, and then she scans one more time to ensure the cavity is completely cancer-free.
So Yeah.
Can I add to that real quick?
Sure. Please do.
So I think, Kevin, you described it very, very well. And just one point for the audience to note is that this has been designed with the surgeon and the current workflow in mind. And so it's adjunctive. And all that means is a physician will do the standard of care like they always do. We come in at the very end, and for less than 10 minutes of time, they do this extra check with the device. So it's not like everything changes around. It's standard of care, and then Lumicell at the very end for less than 10 minutes to get this extra step.
Yeah, so part of the reason why we do this sort of Luminary series interview, even though it's important to understand the what we're raising money for at the pitch, is that it's helpful to understand what are the alternatives that would be standard of care. Let's use the medical terms, and let's answer that question first and then go on to see if there's any competitors. But you demonstrated by some statistics, Kevin, that if you don't get it all, the probability of it getting worse is pretty high, like recurrence, right? So absent this, what is being done to thwart that problem?
So there are a couple things. So I want to start by emphasizing how important surgeons take this, how important it is to get complete resection. Okay, because as you said, I mean, and as I've said before, incomplete resection, cancer left behind, doubles the risk of recurrence. So what surgeons do is they have different techniques that they follow. They may take the primary tumor, they may take a large amount of the primary tumor, but then they may do what's called cavity shaves. So they do cavity, so they take extra tissue,
It's a guess.
Healthy tissue.
Yeah, it's a guess.
Exactly, it's a guess. So they take extra tissue, and that helps them to ensure that the orientation may be correct from a shave versus the primary lump. So they do those techniques. There are other techniques in the OR that they're X-raying the device or X-raying the tissue that's been removed, and they want to see what the edge is, if there's calcification on that tissue. But that doesn't necessarily mean they're catching cancer. They also, in the end, after the procedure, after the patient has left the operating room, they rely on margin pathology or pathology assessment to determine that positive or negative margin. Now, you touched on competitors also. There are multiple competitors. I don't even know whether I call them competitors. There's multiple technologies that are either in the market or being investigated for improving margin pathology. So what they're trying to do is they're trying to bring margin pathology back into the OR so that you're actually examining that tissue better than you're doing, or you're analyzing that tissue while the patient's still in the OR.
So you can go back in if it indicates that you've missed something.
Correct. We had one surgeon in our study that actually stays in the operating room, sends the tissue down the hall to a pathology lab, and has them look at it. The final reading is later after the surgery, but it's just one more check. So there's a lot of different things that the surgeon is doing at the time to ensure complete resection. But let me give you one statistic. And these are the facts around breast cancer surgery. It starts with 20 to 40% of the time, they have to do a second surgery because there's a positive margin. But 65% of the time, when they do a second surgery, they're not finding tumor in the surgical cavity. So margin pathology doesn't have a high accuracy rate in general. Okay, so but I can't overemphasize how important it is to get complete resection. Surgeons know that 65% of the time they're not going to find tumor, but they still do a second surgery because it's that important. Now, here's the other fact. 19% of the time, these are clinical studies have demonstrated that 19% of the time, there's tumor that's completely missed by standard of care. Some surgeons call it a satellite tumor. So let's say at a 12 o'clock orientation of a piece of tissue that's been removed, of a tumor that's been removed, the 12 o'clock orientation has a positive margin. When they go back in and do a second surgery, they'll try to take additional tissue from that orientation. But often there's what they call satellite tissue or skip lesion, where there's tumor actually at the 6 o'clock margin or 6 o'clock orientation that they're not picking up. And that clinical studies have demonstrated that happens up to 19% of the time. And that's where the Lumicell solution really adds significant value, is that the surgeon's able to scan the entire cavity. So in that case, we're picking up that tumor at the 6 o'clock orientation. And I mean, we demonstrated that in the clinical study that was just published in the New England Journal of Medicine Evidence. We found 8% of patients. So in that study, 8% of patients had tumor removed by the surgeon using the Lumicell Direct Visualization System. The majority of those patients had negative margins. So the majority of those patients would have been told they were cancer-free, except that Lumicell aided the surgeon in finding that cancer. And these aren't small amounts of tumor. So surgeons will sometimes tell you that we know we're leaving something behind. We know we're leaving microscopic tissue behind. What was found in the study is we're finding 13, 11, 6, 8 millimeters of grade 2 and grade 3 tumor. There was one patient in the study that was a 36-year-old woman that had, I think it was 2 millimeters of grade 3 tumor. So this is large tumor, large aggressive tumor in a young woman. There are other patients in the study that were over 70 and under the current guidelines, they would not have received radiation. They may not have received radiation. And the patients over 70 years old with 11 millimeters of tumor found. So it's material tumor.
So those lingering tumors that are satellite in nature or otherwise, depending on somebody's makeup, could manifest itself into, real cancer again, right?
It could. It could. And that's what all the statistics point to, that, that's where I say doubles the risk of recurrence. Yeah. That's what all the statistics indicate.
So can we get in the weeds with something here?
Sure.
I'm going to read this because I don't want to get it wrong. One of the participants, Scott, thank you for being here, said, I read your paper. I'm an ER physician and an AI machine learning practitioner, and I'm curious if you could comment on your adverse event rate of 1.72% is 3x the 0.7% adverse event rate of 4 contrast dye.
IV, I think that's IV contrast.
IV, sorry. Yeah, there you go. Thanks for helping. Do you anticipate an FDI ding on this? Also, what tumor marker are you using to light up? And is there enough homogeneity of breast cancer markers to provide robust conclusions regarding your approach to delineate margins? So how'd I do? Shit.
Good.
Brent, you want to take that, or do you want me to take that?
Why don't you take the first part, so the adverse events, and then I can talk about, how the mechanism of action works.
Okay, sure. So the adverse event rate, I'd have to go back and look at the paper again to see the number, but the adverse event rate actually compared well with other imaging agents. We had, compares very well with other imaging agents. We had a number of what would be, I think it was 2 serious adverse events, and those were both allergic reactions in the study. So they were allergic reactions. The patient was treated with Benadryl and completed their surgery. So I think that the safety profile of the drug is very safe. I mean, we're not concerned at all about the interaction with the FDA based on that. And again, one of the things is those were allergic reactions in patients. I know one of the patients had a known allergy to imaging agents. And so it's all done in the in the operating room as well. So we feel very good about the safety profile of the drug.
Yeah, and the way the drug works is it's essentially pan-cancerous. It could be for solid tumors. Of course, we'd have to get approvals and do studies on that. But the way the drug works is it's attracted to the proteolytic activity in and around the tumor microenvironment. So as tumors grow, there's enzymatic activity, proteases that happen, and that's the mechanism of action that cleaves the quencher dye off of our drug, and we're left with our fluorescence. And so that activity, if you will, in and around the tumor, there's a little bit of a margin there, is what you would see when you excite the drug with our probe. And as you see the image on a screen as a physician, the image meaning the fluorescence, you would go back in and take some tissue, scan again, and if you did not see any more red on the screen, you could rely therefore on the negative predictive value, which in our study was 98%, to have a high degree of confidence that you got that out.
It just seems by definition it's better than the post-operative seeing if it's around the edges stuff, right? It's a technical term.
Yeah, and Arthur, I think you're spot on. And what's interesting here is none of that goes away, right? So you get a little bit of the belt and suspenders, right? Yeah. So you still get pathology. You still, if they deem the patient is eligible to go to radiation for that microscopic tumor that may be left behind, they go to radiation. But in addition, right, you get this extra step, which gives a lot of confirmation, and certainly for loved ones that would be in my sphere, we would opt for this.
Yeah. So we have a couple more, but to give Dave an opportunity, if you're ready, Dave, might be a good time to jump in if you're still there. Yeah. So, great.
Yeah, I'm here.
Great. So Dave is driving, so we don't want him to put his video on, and be careful while you're talking. Dave, you want to share with us a little bit of your background and then why you voted with your checkbook here?
Yeah, happy to. So my background is 25 to 30 years, somewhere in there, of forming and building and investing in deep technology companies that have ended up primarily being in technology, information technology fields, some in healthcare, and some more for our field, even in agriculture technology. So long-term builder and investor in companies. In the case of Lumicell, we really took the technology out of the labs at the Broad Institute in MIT years ago. It's over 10 years ago. And this was an opportunity to build a platform company that uses a disruptive approach to delivering a better standard of care. So, with the potential to impact people's lives, and identifying a problem and solving it with innovation. And in the case of this company, it was really partnering with the founding team, two individuals in particular that had lost family members to cancer in a very similar way that we're describing today, which is that their family members went in for surgery. The surgeons didn't get all the cancer, and the patients ended up dying prematurely. And so those family members, that really hit hard. And so we came together with that founding team to start the company. We're long-term investors. We've been a lead investor in this company from inception and are continuing to invest as we go into this Series D financing, a growth financing to support the final approval step here and going into commercial markets. So we're patient capital, and we're really involved. So we've been involved with the company from the beginning. We work with the team day to day. We bring our network and we bring our experience to bear in helping the company. The only other thing I'd leave us with is in the intro is that we've been through these stages of commercialization many times before, and understand the steps and the DNA needed to go from one stage to the next and we think there's a really clear opportunity here to build a meaningful company, both in enterprise value but also in impact. And so we like the fact that it's got both attributes, that we can build a scalable company that's got not only solving problems in the cancer world for breast cancer, but across many other solid tumor surgery areas. And so we have a long road ahead of building enterprise value. But as we do it, we make a bigger impact on the world in an area that's really been in the dark ages and needed to be innovated on. So I hope that helps.
Super helpful. And I was going to ask you about the path to other cancers, and it seems like, one step at a time, but it is essentially the same thing, just different area of the body, right?
Yeah, and I can let Brent and Kevin talk about it more, but the company's been not only in clinical trials in the breast cancer market across many different types of hospitals and teaching hospitals and community hospitals, but also working in other indication areas like peritoneal, bladder, prostate, brain, and others. So there's been quite a bit of work done over the years in other cancer indication areas. And that really goes back to the platform approach to building the company.
Yeah, super.
Yeah.
Thanks so much, David.
Yeah, and I'm happy to share some where we're at on some of those. So we have early stage trials underway in other indications that are really meant to confirm the pan-cancerous approach. I mean, as Brent talked through the mechanism of action for our imaging agent, the imaging agent was designed to be pan-cancerous. So we've studied in prostate, colorectal, esophageal, primary pancreatic, primary ovarian that are peritoneal metastases, and have demonstrated the ability of our imaging agent to pick up tumor in those cancers. And the surgeons were doing most of that early stage work at Mass General here in Boston. I'll tell you, I asked our peritoneal surgeon, I said, how important is it to use the Lumicell system? So I asked him two questions. I said, tell me what you think about the 2 hours before surgery, how that works, and tell me what you think of using the Lumicell solution on your patients. So he said, and again, this is a clinical study, but he said, first of all, I like the fact that 2 hours before surgery fits into my surgical workflow. He said, my patients are sick. And he said, if I have to have them come in ahead of surgery to do anything, it's a major problem for those patients. Okay. What he said next really kind of, was kind of emotional in a lot of ways, because he said, look, when a patient's coming to me to treat for peritoneal metastases, they typically have 3 years life expectancy. He said, but if I leave anything behind, if I leave any cancer behind in that patient is likely to die within a year.
That's
and yeah, and so it was. I mean, I was surprised that he was as broad as that. But that's what he conveyed to me there, and so it does really make a difference. We are treating our GI surgeon, our GI investigator. Came to us and he was very excited because he's using this to treat Barrett's esophagus, not to treat, but he's using it, evaluating it for Barrett's esophagus patients. And he said, that's challenging because some of these patients have to have random biopsies every 6 months or every 3 months. What really excited him was that not only was he able to see the different tissue and target where he took biopsies, but he could see the different grades of tissue. He could see different types of tissue using our imaging agent. So it's again, these are early stage confirmatory trials, but we've gotten great feedback. And the next stage for us would be to complete, to do a larger study in those indications, and that's what we'll do. That'll be in the coming year or so.
So I want to talk about if it's okay, the significance and the importance of the latest information with the New England Journal of Medicine and then the presentation that was made. But let's take one more question from Scott, if that's okay.
Sure.
Have you thought about applying this to MOH's surgery?
Mohs surgery.
Yeah.
I actually, go ahead. I'm sorry.
Which, just to be fair to Scott, I'll finish the question. Surgery, which can be really challenging with tumor margins in skin cancers. And he said, hold on. Correction, I meant to say 2.5x. Okay, this is a correction for the first numbers. So we'll cover that later. Okay.
So as someone who's had Mohs surgery multiple times, I can speak to that. I've actually, I have, as you know, I've had Mohs surgery. I've spoken to my dermatology surgeon about the Lumicell solution for that reason. So I've had it. Look, I think that we think it potentially could work in Mohs surgery for skin cancer. The biggest challenge there is the workflow in that in-office procedure for us. So that wouldn't be one of our initial targets. You know, the in-office procedure, as I've had it done, you go in, you're there maybe 15 minutes before they go in. They remove the tumor, or they remove the skin cancer. You sit in the waiting room, and then they go back in and take another shave if they need to. So there's no IV. So, you know, it wouldn't, you know, we potentially would work in that setting, but it's not our initial, it wouldn't be one of our initial targets because of primarily the workflow in that regard.
Yeah, that makes sense. You're not going to take 2 hours to come in and drink the Kool-Aid or however it's administered.
Yeah, it's an IV. It's an IV injection. So
super interesting. So let's talk about the importance and significance of this latest information for the New England Journal of Medicine and so forth.
You want me to grab that one, Kevin?
Yeah, you want to take it?
Sure. So yeah, it's been a busy and important month for Lumicell. We were honored and thrilled to have our pivotal trial published in the New England Journal of Medicine Evidence. This is a journal that presents innovative, original research, mainly with clinical trial design and then also clinical decision making. So very appropriate with our trial. We also had Dr. Barbara Smith. So she's a lead investigator of ours. She's professor of surgery at Harvard, and she's the director of the breast program at Mass General. She presented the data at the American Society of Breast Surgeons. That was about 2 weeks ago. That happened to be in Boston, where we're based. And she had a packed house as she delivered the talk. And she really concluded 5 things at the end of her presentation. She said, Lumicell helped surgeons find residual cancer after the surgeon said the standard of care was complete, meaning they were done. But then again, as Kevin mentioned, those women that had negative margins, right? So very meaningful that Lumicell then reduced the need for second surgeries due to women who had what would have been positive margins converted to negative with the assistance of Lumicell. That they only took an average of 10 cc's of additional tissue. Of course, that's clinically meaningful. A very high negative predictive value. That means when the Lumicell signal is negative, there's a very high likelihood, in this case 98%, that it is in fact negative, which is a confidence booster. And then the last point she made is that it's got a very nice safety profile, to the question earlier, I think from Scott, and that it integrates very well with the current workflow surgically. So those are the points that she noted that she cared about, and she felt other surgeons may care about too. So those two things together led to a very big, meaningful month for us at Lumicell.
Yeah, that's great. So what's the time frame without backing you in the corner of commercial viability here, assuming all the things?
It's not backing us into a corner at all, because I could just give you what the guidance that the FDA gives to us. So we've submitted our NDA, we submitted our PMA. Mid next week, we should find out whether it will be an additional 6-month review on the NDA or an 8-month review on the NDA. So there's guidelines there. The FDA on the drug side, on the PMA side, it's a 6-month review without a panel, a 10-month review with a panel. And panel reviews on the device side are not that common. So we feel like we have a pretty good opportunity for approval by the end of this year and commercializing in Q1 of next year. Now, if by chance the FDA, because this is a novel product, wants to have a panel review, it's just that it pushes by a quarter.
Yeah, so that's not bad at all.
No.
So, all right, we're going to talk more about this in the next, but let's just touch on the financial ask.
Okay.
This round, what are you looking
Sure.
for? What you've raised?
We're raising roughly $40 million in equity, and considering, I mean, we've had some discussions, opportunities to look at non-diluted debt financing as well. So we're looking at that. But on the equity financing, the insiders are looking to come in around the $15 million range. We are having discussions with med tech investors and strategics that, in the $5 to $10 million range, and we're looking for. I mean, so there's roughly about a $20 million opportunity, and we're having various levels of discussions with other groups. Now, what this does for us is that this really, I mean, this is focused on commercializing in breast and driving to, let's say, an $8 to $10 million run rate in breast. Our financial plan drives roughly $5 million the first year because we're getting started, but then up to above $35 million the second year in revenue. So we're really focused on building to be able to scale up very quickly in that, as well as expanding the platform. I talked earlier about trials and work in other indications, because this is a platform technology. So we're not stopping at breast. We're launching breast, and we're building on that momentum with breast cancer. So Dave, do you want to share anything?
Yeah, go ahead.
Yeah, sure. I'll share a couple more things. So over the last decade or so, we've invested about $100 million in the company to advance through all these stages across all the different cancer markets that we'll end up moving into. Our theme on the company, when you look at how we underwrite this, is we're building a company that is enduring, that will serve a purpose in the market, and therefore be part of the landscape. So we look at this as a company that, sure, it could get acquired in the next couple of years by a larger medical technology company or even a biotech company focused on medical solutions. But it also has the potential to be a standalone business, which is very rare because of their novel technology, their patents, the way they've approached building the solution and the impact it has. We also think that it could be a standalone company that could be very valuable over the long term. So that's kind of the thesis on it. We're very, of course, as you can see, we're very supportive of the company. We're going to continue to invest. I like the traction we're getting with strategic and/or investors in this round. This is a good time to join, given how close we are to commercialization and ramping up the revenue.
Yeah, de-risked a lot of it, right?
Significantly de-risked.
Yeah.
I'd say.
Yeah, super. So
yeah, absolutely.
I was going to ask the typical path for these type of companies. Just in my anecdotal experience, is some sort of exit in the short term, but I can see, Dave, how you could go any way on this, but in this case, but there's often not a standalone company option with a lot of companies, right? It's just, you've got to let somebody write you the big check and move on.
Yeah, and we've done that. Over the years, we've sold companies to the large biopharma or medtech investors, or companies, I should say. And And so when you do that, it's usually because you have one shot on goal. You've got one product. It's a point solution. It's part of a portfolio. But in this case, we can build a platform if we decide to. I think the way Kevin and Brent are building the company, there'll be an opportunity to make that decision. In other words, as a good leadership team, they always create options for the board to consider. And I think the options that will be created in the next year or two will be, does it make sense to do a strategic M&A or to growth finance the business forward? And that decision will probably come as early as the end of 2024, early 2025.
Yeah. Yeah, it's good to hear you're close to commercialization here. Yeah, it's a comforting thing. So super helpful, Dave. Thank you. So we're going to get further into the weeds about the financial opportunity and more about the company. But I hope this has given everybody an opportunity to see at what point the company is and who the team is, and that they are knee deep in this and have been for quite some time. So I super appreciate you guys being here. And then we're going to have a more in-depth ask and financial discussion next week, I think, like soon, right?
Yeah, I think it's next Thursday.
Next Thursday. Great. Awesome. That'd be terrific.
Okay.
Well, that was great, guys. Thank you for doing this. And Dave, thanks for jumping on. Really appreciate it.
Happy to. Yep. Happy.
Thank you, Arthur, and thank you for all who joined.
Great. Thanks so much. All right. Till next time. As I always say, thank you for spending with us. The only thing you can't make more time of, and that's your time. Thank you.
So, thank you, Arthur.
You're welcome.